Functional genomics is actually to understand the disease-causing mechanism and the association of the environmental factors, the genotype and the phenotype of the disease, and therefore to understand the disease-causing mechanism and identify actionable information that will lead up to therapy. We actually leverage multi-omic analysis in various ways, including transcriptomic, proteomic, and all sorts of aligned omic study to understand the disease-causing mechanism. PP1A is a centralized capacity for functional genomic research. And in that program, we established some core research support for single-cell analytic, advanced imaging, rare disease functional genomic laboratory, biomedical proteomics, and stem cell and organoid facilities.
So stem cell medicine works with these remarkable cells that are called stem cells. They are basically our raw material. They are very special cells because they can generate various cell types of your body. So we can use these cells to generate what we call nowadays organoids. They are mini organs that can really mimic the niche of the cell type. For example, we can get mini brain organoids that replicate really well the niche of the brain. We work with the researchers to really pinpoint what their needs are and what diseases they want to investigate. So our aim in the lab is to model disease and generate also large number of human cells so that we can develop new treatments for these conditions. The facility, it's here really to provide this new technology to researchers. We want everyone to have access to stem cells and to be able to grow their favorite organ in the dish so that they can understand different conditions and develop new treatments. Perhaps one of the first service providers in the country to really be able to offer this bespoke service. The funding has really allowed us to do the research and development so we can set up the different mini organs in the dish for the researchers.
The single cell analytics facility is where we examine cells on an individual basis rather than on a population basis. That can be either heroes or villains when we're looking at different types of genetic diseases and disorders. Luminesce Alliance funding has assisted the lab by providing support to buy vital equipment that we need to do the single cell assays that are required. So we've got at least 14 projects from Children's Medical Research Institute and Kids Research. These are all single cell projects that are looking at gene profiles and expression patterns of genes and trying to model various diseases.
We can perform functional assays such as cell-based assays, mRNA analysis, immunocytochemistry, and Western blotting to help determine pathogenicity of variants. Luminesce Alliance funding has provided additional staff members for the Rare Diseases Functional Genomics Laboratory and also funds all our reagents.
So Vector and Genome Engineering Facility, also known as VGEF, is the facility that develops and distributes viral vector tools for use in preclinical studies. And it also undertakes custom genome engineering projects for research teams in Australia and internationally. So the funding from the PPM actually was used to build the facility, to establish this facility over 4 years ago. On average, every year, the facility distributes about 700 viral preparations and performs 50 to 70 genome engineering projects for research teams. Viral vectors are tools that we develop based on natural viruses. They are used to deliver the therapeutic cargo to cells and tissues inside of animals or human patients. The natural syringes or pills. So if you have a genetic disorder, you have to correct the mutated gene in every cell or majority of cells in given tissue. And the way it is achieved is we take the viral vectors, we remove the viral genes, put the therapeutic cargo inside, and then those viruses then can go and deliver the payload to every single cell that has a mutation inside.